Now components of yak. So it has certain like so many elements inside it. It means it is made of so many components. So now for the first component that we have is CEN, that is SAN. It is a yeast centromere. It means it's like the chromosome of yeast contains in it centromere. We already know that human DNA also contains so many centromeres. Like centromere is like a protein which binds the DNA in such a way that it looks like a chromatid. It has two partitions. A centromere, that is SAN, is centromere that ensures chromosome partitioning between two daughter cells and the selective marker genes. So, what its function that it ensure whenever the chromosomes are partitioning, whenever the cell is dividing into a two daughter cells, that ensure that chromosomes partition take place and act as a selective marker genes that ensure chromosome partition between two daughter cell partitioning and a selective marker genes. RC, that is autonomously replicating sequence. This is a sequence which replicate itself autonomous. It has not needed any um, ovary like thing, so it gets replicated. It's autonomous. Now we have TAL, that is telomere. These are simple, like just in humans we have the at the endpoints we have the telomeres. Selective markers such as URH3 and TRP1 and bacterial selective markers. So you can see the structure here. Here you can see this is yak. It has restriction sites to which restriction can cut. Then we have centromere, then we have RS, then we have E selective markers, TRP1. Bacterial selective markers are also there because this is going to be replicated in both in yeast as well as in bacteria. So we need selective marker for both. It has again one another restriction site. We have TEL, these are the yeast telomere, then we have E selective markers, another one. So yes. Now how this yeast artificial chromosomes gets construct. Our yeast artificial chromosome is built using a initial circular DNA plasmid, which is typically cut into a linear DNA molecule using restriction sign. We already know that the plasmid, which is double stranded and circular in nature, when it gets cut, when we cut it, it come out into a linear DNA molecule. Okay, and when it turn out to be a linear DNA molecule, then what happened? DNA ligase is then used to ligate a DNA sequence or gene of interest into the linearized DNA. The DNA gets converted into linearized DNA. Now this linearized DNA, we trying to insert our gene of interest and then we trying to ligate, forming a single large circular piece of DNA. Then we construct a single. Already it is circular, then we cut, we add our gene of interest, then it can turn out to be an. You can see here this is your normal plasmid having all these genes like gene for centromeres, for ARS, cloning DNA, gene 3 telomeres are there. We cut it from that onwards, and from that we can cut cloning DNA. This is the site from where we are cutting, and we are adding this 1 MBs, that is 1000 kb uh, gene of interest. Clone insert linearized vector. We cut it, then it converted to a linear. We are adding this again, and then it's converted back into a circular. Cloning into is artificial chromosomes. So, how this is artificial chromosomes cloning take place? So, this is human DNA. We already know it. Chromosomes is Icori partial digestion. Large chromosome fragment take place. Sticky are forming. Now we have GAC vector. This is one, and alright, tell Bamach1, San, Korai, this is the East artificial chromosomes vector. Now we are cutting it with the same restriction gene like EcoR first and EcoR first. Let me back. So yes, we cut. It also formed the sticky ends here. and now we are trying to ligate the recombinant DNA will form. Like we are like DNA ligase is used to ligate these fragments. So in this case, the gene of interest is human genome. So because these are going to be used for the human genome library functions for cloning them, so we just cut here, and then we are just trying to add this human genome sequence or nucleotide inside this yeast artificial chromosome vector. Now it's get ligated and the yeast cell gets transformed. Now whenever the yeast cell will divide, this DNA replic of human genome also get divided with this yeast artificial chromosomes vector. Now it's cloned into so many sequences. Now, what are the applications of yeast artificial chromosomes? A properly modified yeast artificial chromosomes can be utilized in many different organisms for cloning or genome analysis. If we wanted to analyze any particular genome, so we can use yeast artificial chromosomes to let it get inserted with this genome and it gets cloned. And after cloning, the sequence we get, we can analyze this. Chromosomal translocation. Translocation it's what it means. We need to know this. Chromosomes abnormality that occurs due to rearrangement of parts among non-homologous chromosomes. Chromosomal translocation, whenever the sequence of DNA are just getting disturbed, it means they are changing their position. So what can, if you wanted to study this, can be studied by means of disposable yaks that do not contain genetic information necessary for cell function. So we can use yak for studying them. Yak generally do not contain genes for cell functions important. To identify essential mammalian hormonal sequences necessary for the future construction of specialized mammalian artificial chromosomes that we are constructing. And to construct this like mammalian artificial chromosomes, we need to identify mammalian chromosome sequence. To identify this sequence, we use yak. Truly speaking, I'm not interested, but I'm reading and I'm learning so I can get up better. Comparison to Yak and Back, we can see here. Configuration: Yak are linear in nature, Back are circular. Features: host: Yak has yeast, Back has bacteria. Copy number 1, which has 1 to 2. Cloning capacity: unlimited, known to low. Symmetrism 40%. Insert stability: if the insert which is we have placed inside it, that is very unstable and that is very stable. References: you can check out those references for your studies as per you want to. If you have any query or any suggestion, let me know in the comment section. Till then, bye. Keep learning, keep pushing, and bye. Have a good day. This is how it's going. It's great to know with so many things which is needed to be known. Till then, bye. I'm going reading this